Previously, already chemistry in daily life videos are uploaded. You can watch it. I will give the link in the description box as well as in the I button present about this video. So first starting part is that what are the key questions, that is what are proteins, what are they made of and how are they synthesized. So first we are going to see that what is protein. Proteins are basically some complex compounds. These are tagged as macromolecules which are generally made up of carbon, hydrogen, oxygen, nitrogen and sometimes sulfur. They are versatile and abundantly available in organic molecules in the living system. So what are the general characteristics of these proteins? Now when we talk about these proteins, these were first discovered in 1838 by Mulder and named by Berzelius. These are generally colloidal in nature and the molecular weight varies in a huge range, and the isoelectric point, that is Pi, it is the pH at which protein has no net charge, affects the solubility and mobility of the protein. Hydrolysis: in acidic and enzymatic conditions, proteins yield amino acid. Amphoteric, it is tagged as amphoteric due to the presence of both acidic and basic group. Acidic group is COOH whereas basic group is NH2. Solubility, it is based on the solubility differentiated into two types, fibrous protein as well as globular protein. So what is the role of amino acid in protein? This is the basic building unit of protein and that's why it is so important. They won't carry out their intended function without the amino acid being set in order. Next, so amino acid structures are generally made up of, they have a hydrogen atom, an amine group, a carboxyl group and a side chain which varies. So this particular portion requires to know what are the amino acid and how they are classified. Now the detailed discussion of amino acid will be made in a separate video. Here we have only the structures and the essential, non-essential, acidic, basic and neutral amino acid categorized. So valine, leucine and lysine is your essential. Non-essential includes your alanine and glutamine, and acidic and aspartic acid, glutamic acid included in acidic medium. Basic ones are lysine, arginine and neutral is glycine and serine. So what is the primary structure of protein? There is a specific sequence of amino acid in a polypeptide chain and that dictates the protein's properties and biological functions. It is determined by the DNA coding via mRNA. And the example is that insulin has two chains A and B which are present in a fixed sequence. Any change or mutation in your primary structure can lead to disorders. The next part is secondary structure. So regular folding patterns due to the presence of hydrogen bonding between NH and CO groups produces these secondary structures. The types are alpha helix, right-handed coiling and the beta pleated structure. Extended sheet-like structures may be parallel or antiparallel, stabilized by interchain hydrogen bonding. Examples of these ones are fibroin has beta sheets. Next type of structure is 3D structure, tertiary structure, where the polypeptides are being folded in such a way that they form a globule. So maintained by hydrophobic interaction, disulfide bridges, ionic bonds and the hydrogen bonds present in it. Examples are myoglobin, a compact globular protein that generally stores oxygen. The biological activity is being determined by these structures. So active site for configuration in enzymes. Next we are going to see the ultimate structure present within the protein, that is quaternary structure. So two or more polypeptide chains, when they come together, associate together into a functional unit, then it is quaternary structure. The subunits are held by non-covalent interactions or disulfide bridges. An example of such a quaternary structure is observed in hemoglobin which is present in our blood. Important for allosteric regulation and complex function. Example is catalysis. Now we see classification of proteins. Now based on composition, simple, conjugated and derived proteins are obtained. Now in simple ones we have only amino acid, whereas in conjugated ones we have protein with prosthetic groups. Metalloproteins, glycoproteins and lipoproteins are examples. In case of derived, from the breakdown of other proteins, these derived proteins are being obtained. Now based on the shape, they are differentiated into fibrous and globular. Fibrous are long rod-like structure. Structural roles, they have significant structural roles and they are insoluble in water. Examples are collagen and keratin. Globular, spherical, functional roles, enzymes, hormones. Water soluble, hemoglobin and enzymes are the examples. So now the next topic of discussion is denaturation of protein. So when the proteins get unfolded, their structure and function gets lost. Causes is that heat, boiling egg white. This can be one of the main reason for the denaturation of protein. pH change, gastric acid denatures pepsinogen. Organic solvents, alcohol, acetone, heavy metals can cause also denaturation. Reversible, sometimes upon removal of the denaturant, this occurs. Irreversible, most biological cases, and the consequence is that the enzymatic activity of that particular protein is lost. Its function is also lost. So how can we analyze that our sample contains protein and test it? So biuret test is one of the method where peptide bonds generally form violet complex with copper 2+ violet color, and xanthoprotic nitration of aromatic rings produces yellow color. Ninhydrin test, free amino groups form purple color, that is blue or violet can be the coloration. Electrophoresis separation by charge or size in the electric field, we are going to observe band patterns. Spectrophotometry, UV absorbance at 280 nanometer due to tyrosine and tryptophan. Here we can quantify the concentration. Next one is protein synthesis overview. So generally the synthesis process is a complex one. First, DNA is converted into mRNA in nucleus. This mRNA synthesizes protein on ribosome in cytoplasm. Then initiation occurs. Ribosome binds to your mRNA. Then elongation. tRNA's function is to transport the amino acid to it. Termination. Stop codon ends the process of preparing this particular protein chain. So post-translational modifications are there can be folding, cleavage, phosphorylation, and enzymes involved in this case are RNA polymerase and peptidyl transferase. So this is the total overview of synthesis of protein. And the last thing is that what are the recent advances which are included within your protein. That one is AlphaFold DeepMind, predicts the protein folding with high accuracy. CRISPR associated proteins used for genetic editing. Biosensors generally used in reactions. Protein based sensors for detecting glucose toxins. And protein vaccines. COVID-19 Novavax is spike protein based. So these were the general introduction towards the protein, and the topic is being discussed elaborately. In the next video we shall deal with amino acids in details. Hope this was helpful. Thank you for watching. Do not forget to like, share, subscribe and comment.